I will talk about our work on interactive visual analysis of PubMLST data. The dataset in PubMLST are very large, heterogeneous, multidimensional, and complex. It is thus very challenging to assimilate and analyze these datasets using conventional approaches. We need an easy and intuitive means to communicate this data to users of all types and levels of expertise so that they can explore and analyze the data to derive knowledge and gain insight. Our target users are the general public, policymakers, epidemiologists, researchers, and many others. The approach we use to address this challenge is called storytelling. Everyone likes a story. Storytelling is a very good way of communicating information. It gives a high-level contextual narrative view of what is happening. Our storytelling is achieved using visualization and visual analytics. What is visual analytic? It is an analytical reasoning approach supported by highly interactive visual interfaces, which can support us to explore and exploit the data to answer the W questions, which are what, where, who, when, and which, so that we can understand the why. We need to transform the inherently non-visual data into visual form that is intuitive so that users can do their work easily and efficiently, such as to detect trends, patterns, and anomalies, being aware of the situation, support their decision-making, as well as discovering knowledge. Here are 4 examples of our storyboard: Campylobacter, Klebsiella, Neisseria, gonorrhea, and meningitis. I will run through some examples of our storytelling systems. First, I would like to tell you about our Campylobacter storyboard. At the top of the page, we provide an introduction to Campylobacter. Here we show a summary of some data from our database. On the left is the gender information, and on the right is the temporal pattern. Here is some summary information about Campylobacter, and this area gives information on precaution to prevent Campylobacter infections, while treatment information, publications, and PubMLST data can be accessed by clicking on these panels on the right. Here on the left, you can click to drill down and explore the age group of the patients, their gender, and Campylobacter species. Let us click on the age group panel. Here you can interactively explore the age group of the data in PubMLST. For example, this is the Sankey diagram, and it shows the relationship between the age group and the gender. By moving and adjusting the time slider, you can see the number of infected males and females during the selected time period, and the most recent 5 cases here. Similarly, you can explore the relationship between clonal complex and the gender, and the relationship between clonal complex and Campylobacter species. Here, clicking on any of the information boxes will take you directly to PubMLST to access the isolate information. Now let us look at our Neisseria meningitis storyboard. Once again, the top panel provides an introduction. The middle shows the summary of the geographical and temporal information for the data from PubMLST. And once again, here are the information and exploratory panels. The supplementary information panel gives you more information about Neisseria meningitis, and the additional information panel provides information on how it spreads. Publications and data can be accessed through the publication and data panels. You can also find out about the dataset, as well as symptoms and treatment. And finally, by clicking on the age group and region panel and the serogroup and clonal complex panel, you'll be taken to the drill down pages where you can explore the data. Let us click on the age group and region panel. The age group of the patients and their geographical distribution is important in assisting the public health response to the disease. In England, like many high-income temperate countries, most disease is in young children, with elevated levels also seen in young adults and the elderly. It is nationally distributed, with some variation in rates in different regions. Here, users can explore and analyze the relationship between age group and the 9 geographic regions in England and their associated serogroup and clonal complex by using the drop-down filters for age group, region, and epidemiological year. For example, users can explore the Northeast region during July 2012 to June 2013. Again, this enables users to explore the relationships, patterns, trends, and anomalies. Users can also analyze the relationship between serogroup and clonal complex and their associated age group and geographic region by filtering on a specific serogroup and clonal complex. Now let's just look at our Klebsiella storyboard. Here we talk about defining Klebsiella species using RMST. This will take you to the bacterial speciation story, while this will take you to the data visualization drill down. This takes you to what we call the explainers. This is the explainers page. As you can see, if you want to find out more about, for example, Klebsiella, its species, or what is RMST, by clicking on any of the boxes, you can find out more. Let's click on Klebsiella type strains. This will take you to the Klebsiella type strains page. Now we can explore the data. This is called a box plot, and it shows the 17 Klebsiella and related Rotella species. Brushing on any of the 17 species will show you its minimum, maximum, and median size information. You can also change the display to explore the data in a different manner. For instance, this is called a violent plot. As you can see, you can choose different ways to display data so that you can explore different aspects of the data. In summary, our web-based storyboards enable users of all types and levels of expertise to explore and gain understanding of the information contained within PubMLST in an easy and yet detailed manner, without the need for a user manual or any training. We'd like to acknowledge the support from the Wellcome Trust, the Food Standards Agency, Public Health England, and the National Institute for Health Research.